Thank you so much, and thank you all for attending on this last day, and especially for choosing to enjoy the free air conditioning here as opposed to the airport. I really appreciate it. So I'm Sue Ishak, and I'll be presenting the microbial data, which is part of a much larger study by Elena, who's the graduate student, James, and Brian out at University of Illinois, looking at calf health in the first few weeks of life. So to summarize several decades worth of important research into one very small slide, we know that high quality colostrum is incredibly important to the developing neonate, especially in young calves. It promotes epithelial function, host metabolism, immune development, and of course microbial colonization. And then we also know that microbial colonization is influenced by numerous factors, including the birth environment and local factors such as temperature, pH, and then, of course, host epithelial types. So in this study, we looked at the impacts of colostrum on the choreography of neonatal digestive tract microbiome, and specifically today I'll be talking about the bacterial microbiome. So we chose 12 healthy male Holstein calves. They were separated from their second lactation dams immediately after birth, and of course given a full physical exam and checked for health. They were fed 4 liters of aseptically collected high-quality colostrum. That quality was determined by a refractometer, and that colostrum was from their respective dams. They were fed 4 liters immediately, and then again 4 hours later after that colostrum had been refrigerated for 4 hours at 4 degrees C. The calves were housed, monitored, and fed separately for up to 21 days. After those 2 initial colostrum intakes, they were fed on a non-medicated commercial milk replacer. And then 3 calves were euthanized, each at days 1, 3, 7, and 21. Now for our sample collection, we collected from these dams on day 0. We collected colostrum. We tested it fresh and then again at that 4-hour time point. We also took udder scrapings and then vaginal scrapings. And for our calves, on days 1, 3, 7, and 21, we took pharynx scrapings, and then we took digesta and epithelial scrapings from the rumen, reticulum, omasum, abomasum, duodenum, 3 locations within the jejunum, ileum, and colon, as well as fecal digesta that were taken on a wider variety of days. From there, we used Illumina-MiSeq of the V3 to V4 region of the 16S rRNA gene from all samples, and this is where I came in. So I assembled contigs using PandaSeq with default quality parameters and an overlap of 15 nucleotides. And my analysis was primarily done with mother. I quality called sequences which were not the right length, anything that contained ambiguous bases or homopolymers over a certain length, identified as chimeric, by UCHIME, or anything that did not align or classify to a Silva 16S database, and if that's all gibberish, don't worry about it. The important thing is I only took high-quality sequences, and then I subsampled them down to 5,000 sequences per sample for my statistical analysis, which was done using mother, R, and primer. So suffice to say, there were a lot of results to go over, and in the interest of time, what I've done is highlighted only the 3 most prevalent phyla, which happen to be Firmicutes, shown in green, Bacteroidetes, shown in purple, and Proteobacteria, shown in blue. Suffice to say that in all calf locations, diversity did increase over time. So over here in our first column, we have our maternal samples, and then along the rest of the axis, we have our calf samples that are laid out proximally to distally along the digestive tract. So we have rumen over here, and then all the way down to our fecal samples. So what we saw is that Firmicutes, again in green, was increasing as you went proximal to distal along the digestive tract, whereas Bacteroidetes, shown in purple, and Proteobacteria, shown in blue, tended to decrease as you went along the digestive tract. If we look at those same digesta samples over time, so from our time point on day 1 to day 21, I've sectioned them all out into location. We can see that Firmicutes, again in green along the bottom with these arrows, had a tendency to decrease in the rumen over time and increase at the lower intestines over time, whereas Bacteroidetes in purple, shown in these arrows here, had a tendency to increase in locations over time. And then Proteobacteria up here in blue had a tendency to increase in most of the rumen, not all of it, and decrease, again, by that lower intestine, jejunum, the distal jejunum, ileum, and colon. When we looked at the calf scraping samples, we saw that a lot of the players were still the same, but their abundance was drastically different. Again, if we're looking at that digestive tract from proximal to distal, we have our maternal samples here. We have our pharynx scrapings up here, and then we go from the rumen all the way down to the colon. We saw that Firmicutes had a tendency to be increased as you went through the chambers of the stomach, but it decreased along the small intestines. Whereas Bacteroidetes, as we saw with the digesta, had a tendency to decrease as you went proximal to distal. Proteobacteria was increased as you went proximal to distal, and it was interesting that it was greatly increased in our mucosal scrapings over our digesta samples, possibly due to oxygen scavenging from potentially pathogenic species. Again, if we look at those calf scraping samples over time from day 1 to 21, we've got our maternal samples, our pharynx, and then our intestinal tract, or our digestive tract. We saw that Firmicutes in green had a tendency to decrease over time, whereas Bacteroidetes in purple increased over time, and that Proteobacteria also decreased over time. So if we look at all of our individual samples together on a non-metric multidimensional scaling plot, or NMDS, we saw that location along the digestive tract was the strongest predictor of variation and how our samples would cluster out. So I've color-coded them rainbow colored as we go proximal to distal, with the maternal samples in black or gray, and then anything that is open is a scraping sample, and anything that is filled in is a digesta. And we see that here in our reds, oranges, and yellows, we have our primary stomach sample clustering, so everything from the rumen through the abomasum. And then in this somewhat wider cluster, we saw that most of our intestinal samples were clustering together, and then the maternal samples tended to cluster together. So we also did see effect by day, which is to be expected, although it was a less strong predictor than body location. And we also saw an effect by type, digesta versus scraping, but it was not as strong as we had expected at first. So that was sort of a lot to take in. So what I did was I selectively colored my body locations and then took a bunch of screenshots and assembled them into a crudely made GIF, which we'll hopefully play, so that we could more easily look at what was happening as we went through the digestive tract proximal to distal. So what we saw is that our colored samples over here were becoming more similar to maternal samples as we went proximal to distal along the digestive tract. Let's show that one more time. So as we go through the stomach and then into the intestines, we just generally see this shift closer to the digestive or the maternal samples. If we separate that out and concentrate just on our colostrum samples shown here in gray, and now I've selectively colored them by sample day. So we have days 1, 3, 7, and 21. I've blotted out any of the fecal samples that were from those alternate days because we only had fecal samples from those days. And what we see again is that our, come on, you can do it. There we go. Our calf samples are again becoming more similar to colostrum over time. Just a little harder to see because it's faster. And then using Bray-Curtis dissimilarity, we found that the most similar calf samples came from the duodenal samples, the medial jejunum samples, ileum on day 7, and then our fecal samples. And then you're in luck. I did have time to analyze not only the udder scrapings, but the vaginal scrapings, which is not reflected in my title. But we saw that that was reflected in both of those locations as well, that over time you are seeing those calf samples become more similar to udder scrapings over time. And in the udder, our most similar calf samples were from the abomasum, the duodenum, and the feces, again also by Bray-Curtis similarity. And then one more time with those vaginal samples shown in gray is that they are clustering more tightly to those vaginal samples as the day proceed. And our most similar calf samples to those vaginal samples were the proximal jejunum, the colon, the duodenal scrapings, and then again the feces. So what I've done here is selected a small portion of a correlation plot where our positive correlations by Pearson's are shown in blues and our negative correlations are shown in red, and the intensity of the color is indicative of that correlation score. We saw that body weight, but not specifically day, because of course in neonate animals body weight is usually positively correlated with day. But specifically with body weight, we had a number of genera that were positively associated, and then one family which was negatively associated with body weight. And then in terms of somewhat related health parameters, including rectal temperature, heart rate, and respiratory rate, we again see that there were several genre or families which were positively or negatively associated with each of these factors. And what's interesting is that we do start to see some of our potentially pathogenic genre show up, such as Villanella, Klebsiella, Escherichia, Shigella, and then also a few of our genres which we consider to be more protective, such as Lactococcus and Butyrococcus. So our major conclusions from the study, as presented here, was that the digesta and the mucosal microbiota were indeed different based on their abundance, but not necessarily on their players, and they were not statistically significantly distinguished by 21 days, which is very interesting because they present two very different environments. This was determined by linear discriminant analysis, which showed that there were 0 OTUs or taxa which were discriminatory to either one of those locations, anywhere along the digestive tract. Also by Permanova-Amova, which is analysis of molecular variants, and then analysis of similarity. We did see that our calf samples progressively resemble our maternal samples in all 3 locations over our 21 days, which is not surprising. It's likely that the maternal bacteria had time to colonize over those 21 days, and so as we progress, they tend to have a better hold on that digestive tract. And then, of course, the external environment is unsuitable to in utero bacteria. So typically, the in utero environment is thought to be sterile, but of course, pathogenic species don't like to play by the rules, so you never quite know. And then, of course, functional development along the digestive tract, such as the growth and development of villi, and local changes to the digestive tract, such as pH, temperature, that sort of thing, of course, do drive microbial selection, so that's a lot of what we're seeing here. And these results are fairly consistent with other studies that look at the development of neonates in the first few weeks of life. So jumping off from there, I just got the immunological data back from the rest of the group, and I haven't had a lot of time to analyze it, so I apologize for not being able to present it here, but if you're willing to contact me afterwards, I can give you a few hints. But interleukins, interferons, TNF-alpha, and then several toll-like receptors were also measured in various sites in the body, specifically in the intestines, and right now we're correlating them to not only diversity but also specific taxa in the intestine. So we see that there were spikes at certain days and in certain body locations in our different immunological values. So can we tie that in to changes in Klebsiella or something like that? The short answer is yes, and it is very specific by body location and by day, which is why it's taking a little longer to tease out. So that was a small part of all the information I have. So if you do want more information and more specific information on the rest of the calf health study, we do have some publications coming out soon. You can easily contact me. I'm all over social media, and you can also contact any of the other co-authors who would be happy to answer any questions. And with that, I will take questions here. Don't be shy. Thank you very much.